When presented with a bacterial infection, medical professionals often prescribe antibiotics as a conventional method of treatment. However, overuse and misuse of antibiotics can lead to the development of antibiotic resistance, causing adverse effects to human health and making future infections increasingly difficult to treat. This is particularly evident in the bacterium Clostridium difficile, an opportunistic pathogen within the human GI tract that leads to antibiotic-resistant infection after antibiotics are used to treat another condition. C. difficile is listed by the Center for Disease Control as the leading cause of antibiotic-associated infection and one of the most common healthcare-associated infections in the United States. It's even listed in the CDC's Emerging Infectious Disease Surveillance Program, highlighting the need for novel treatments to combat this specific infection, as it really affects healthcare workers and those patients above 65 years of age. This in vitro experiment seeks to answer how the treatment trajectory of C. difficile infection will be affected when utilizing inorganic nanoparticle therapy, in comparison to conventional methods of treatment using further antibiotics. Current methods of treatment for antibiotic resistance are insufficient and continue to exacerbate the cycle of antibiotic use. Because of the complexity of this issue, antibiotic resistance requires an interdisciplinary One Health solution. The One Health initiative connects the health of people with the health of animals and the environment we coexist in, drawing in professionals from these fields. By utilizing inorganic nanoparticles to target antibiotic strains of bacteria, we circumvent the use of further antibiotic treatment and implement a technology that sits at the intersection of many disciplines within science. This experiment uses the sol gel method to expose C. difficile to nanoparticles. Based on existing research, inorganic metal oxide nanoparticles are expected to be a more effective treatment for C. difficile than antibiotic alternatives.
Our question for this is, how is treatment trajectory of C. difficile infection affected when using inorganic nanoparticle therapies as compared to conventional treatment using vancomycin? It is hypothesized that the use of inorganic nanoparticles as a treatment for persistent C. difficile infection will decrease overall treatment time of the infection as compared to the conventional treatment with regular antibiotics.
This experiment considers 4 different nanoparticles: silver, titanium dioxide, magnesium oxide, and superaromatic iron oxide or spion. Silver is one of the most common metal nanoparticles. Its medicinal qualities known for at least 2,000 years. Observations in modern experiments, however, show that silver can cause pitting in cell walls and can inhibit protein synthesis. Similarly, titanium dioxide has also been well studied. Titanium dioxide is photocatalytic, responding to visible and ultraviolet light, causing a burst of reactive oxygen species, damaging cell membranes and DNA. Magnesium oxide is known to be efficient against bacteria and is already used in economically viable products, despite being less well researched than the previous two. It is effective against viruses, spores, and gram-positive and gram-negative bacteria, and may prevent biofilm formation. Unlike the last three, spion is a relatively new nanoparticle and has not been researched significantly. It could present a new approach to nanoparticle treatment not yet exhibited in any other. Researchers use spion and magnetic fields to cause localized hypothermia in damaged cells.
So, in order to look at this experiment and how treatment trajectory is affected, we use in vitro methods and bacterial plating to compare the effectivity of different nanoparticles to create zones of clearing in the C. difficile growth. First, each of the nanoparticles are prepared using the sol gel method specific to each nanomaterial. After they're preparing, we are inoculating CCFA plates with C. difficile using sterile technique. These CCFA plates provide the proper nutrients and growth conditions for C. difficile to create a lawn of growth on the plate and allow us to inoculate with the prepared nanoparticles in the next step. We inoculate the prepared nanoparticles in the center of the C. difficile plate and compare it to a control plate of a vancomycin disc. From then, we will allow the plates to incubate in aerobic conditions similar to the gastrointestinal tract for 48 hours or until a complete zone of clearing has formed in all plates. The plates will be recorded with a camera in time-lapse mode with a ruler and frame for us to measure and see the rate of clearing formation. After the data has been collected, we will compare the rates and zones of clearing formation from the experimental plates to control plates. Tying into our hypothesis, we hypothesize that in all of the experimental plates with the inorganic nanoparticles, they will have quicker rates of zones of clearing formation and larger zones of clearing as compared to the control plate with the vancomycin disc.
So we can conclude that with successful results, we can continue further studies on the best performing nanoparticle and to start performing in vivo studies using mice models as a precursor to human clinical studies. We plan to study how the specific nanoparticle will perform within the GI tract of an organism that has C. difficile infection. By introducing this method to in vivo studies, we will be able to further explore the biocompatibility of the nanoparticles and analyze the toxicity concern of the nanoparticle. Ultimately, this will help determine proper dosage for in-human treatments and inform other studies looking to use nanoparticle therapy for different antibiotic-resistant bacterial infections.